Hello, in this session we'll plan to discuss the Heamatoxylin and Eosin dye. We shall also discuss the prerequisite steps required prior to staining to enable dyes to permeate tissue. We will then discuss the steps after staining to make the preparation and staining permanent. We will discuss quality control and the results of the Heamatoxylin and Eosin dyes applied correctly. When we are staining for histology, the tissue section is applied to a glass slide. The tissue section sticks to the glass slide using a static interaction. These slides with tissue sections on them are ready for staining. The slides are introduced into an oven or heated device. This oven or heated device will be at between 60 degrees centigrade to 70 degrees centigrade. The time that the tissue sections are baked on to the slides varies according to the tissue type. Routinely, this is a shorter time. If the tissues are specialised, for example brain or undecalcified bone samples, these samples are baked on for longer than the routine sections. Once the sections are baked on, the paraffin will look dripped off the tissue and microscope slides. This can be seen by eye. These slides are now ready to be cleared in xylene. The xylene removes any residual paraffin and cleans up the tissue. The slides are then placed in graded alcohols with the highest, strongest grade first, so for example near 100% alcohol, then going down where more water is in the alcohol. This is the down to water step. The slides are then placed in water ready for staining. The reason why slides are placed in water is because our dyes used routinely in specials are usually made up in water or water-based solutions. The slides are then stained as per recipe or method in the laboratory, and then once the slides are finished with the staining, they have to be made permanent. We do this by taking the stained sections up through graded alcohols, so from a watery alcohol to a near 99% solution alcohol, through xylene for clearing and drying, and then mounting with a mountant with the same refractive index as the glass. So we use a cover slip, the mountant glue and the glass slide with the tissue section. This is a permanent preparation. Then these slides are ready to be quality controlled and digitized using a scanner. These preparations are permanent and the dyes are less likely to stream or move out of the place that they are intended to be demonstrating the tissue elements we require. So the Heamatoxylins are actually a natural dye. They are from the logwood tree, which is native to Central and South America. The tree is the Heamatoxylin campicianum tree. The extract from the tree is oxidized. This is called hematin. This oxidation of hematin is achieved in the laboratory by either a natural means or a chemical means. The natural means is the hematin and dye solution is left in open air for it to mature. This oxidation is important prior to staining because it makes the hematin effective. The chemical maturation is achieved using For Harris's Heamatoxylin, mercuric oxide. For the gills, it's sodium iodate. And these chemical oxidizers mature the dyes quicker, but they aren't without their limitation. So using chemical oxidizers can force the dye to reach its ripened state quickly, but it can also deteriorate quickly. And this is why we have to quality control our heamatoxylins daily to make sure that there are appropriate effectivity of staining for the heamatoxylin and eosin stain for that day. If the heamatoxylins are deemed ineffective, then the solutions have to be replenished with ripened heamatoxylin dyes. We can use an alum heamatoxylin or a mordant to make the heamatoxylin staining more permanent. This is done with, for example, the Weigert's heamatoxylin. The mordant we use is an iron salt, which is usually ferric chloride. This ferric chloride mordant makes the heamatoxylin permanent, and this is essential for when we are using strong chemicals after the heamatoxylin that could wash out the heamatoxylin, such as picric acid in trichromes. These mordants are a substance which is an intermediary between the basophilic tissue that we're trying to stain, for example the nuclear material, and the dye itself. These intermediate substances can also be called a dye lake. So there are natural dye lakes present in tissue, for example in undecalcified tissue sections that have calcium. Calcium stains with heamatoxylin a pale blue. There's no detail to the substance, but you can recognise it's calcium as it takes on a blue colour. This mordant tissue complex is virtually insoluble in most fluids, and the mordants are trivalent metals such as iron. And this is a cationic metal binding to both the dye and tissue. After the heamatoxylin has been placed, we check for differentiation of the heamatoxylins. Some heamatoxylins are progressive, so they're placed in time stages to reach the maximum point of permeation of the pieces of the tissue we want to demonstrate. So for example, we'll stain with heamatoxylin until the chromatin is seen in the nuclei to a high extent with a good contrast between the non-dyed tissues. We can also use heamatoxylin regressively, which means we overstain with heamatoxylin and then we remove a little of the heamatoxylin to produce a contrast between the stained nuclear elements of the tissue and the cytoplasmic elements of the tissue. So this acid alcohol treatment removes a little of the heamatoxylin we've put in previously in order to allow the cytoplasmic elements to be stained and a contrast and distinction be made between nuclear parts of the tissue and cytoplasmic parts of the tissue. The cytoplasmic stain we use routinely is an eosin. An eosin is an acid dye carrying a positive charge which will attract negative charged cytoplasmic elements within cells. Eosin Y is used commonly. Historically, we used to use Eosin Y in water with a little bit of phenol to prevent any bacterial or fungal growth. Fungal growth actually. We now use around a 0.5% solution in alcohol. This will stain the tissue elements, for example, cytoplasm of the cells. It'll also stain fibrin, red blood cells, and also any keratin. Collagen will also be stained. So the resultant Heamatoxylin and Eosin well-stained preparation should have nuclei and RNA-rich cytoplasm and calcium staining blue or navy blue. Muscle fibrin keratin would be red, collagen will be pink, and red blood cells will be an orangey red with a birefringence. These preparations are quality controlled and then submitted to the reporting pathologist or specialist reporting scientist. These chromatin and nuclear details are particularly important when determining whether a cell is cancerous, precancerous, undergoing repair, transformation or apoptosis, or if the cell is in a normal state. The nuclear detail is an essential component of diagnostic importance. We look at the nuclear envelope, we look at the chromatin detail and the lack or presence of a nucleolus to make a diagnosis. The cytoplasmic stain is particularly useful in differentiating cell type. So if a tumour is well differentiated, we can make a distinction between cells present. And all of these features should be demonstrated well with good contrast on the prepared slides. Thank you for listening. Enjoy staining.